Okay. So hello, my name is Nikita, and my project is about interaction between gamma and beta oscillations in the cortex. So usually in the cortex, gamma oscillations appear when a new information is processed. And at the same time, beta or alpha oscillations decrease. And one of influential hypotheses about this is that beta oscillations promote cortical status quo, which means that beta oscillations tend to stabilize cortical codes from being changed. And as opposed, gamma oscillations serve for delivering, transmitting new information in the cortex and changing states of local populations. And usually there is an anti-correlation between gamma and beta power. For example, here is a working memory example. And as you can see, when stimuli are presented, gamma oscillations are stronger and beta oscillations are suppressed. And on the contrary, when the information is retained in working memory, so you don't have to update your state, your code, beta activity is strongly increased. Okay. And it is known that gamma oscillations are generated in superficial cortical layers, and beta oscillations are generated in deep cortical layers. And there is a beta band drive from deep layers to superficial layers. And it seems to be that this drive is inhibitory. So the idea is that we have some sort of beta generator in deep cortical layers, which inhibits gamma generation in superficial layers. And making this, it makes it insensible to gamma band inputs, and thus it prevents loading or updating of information in the circuit. And this is basically what I want to model in this project. Two simple oscillators, excitatory inhibitory, layer 2, 3, and layer 5. Layer 5 is supposed to generate beta oscillations. Layer 2, 3 is supposed to generate gamma oscillations. And if we deliver gamma band input to L23, it will entrain this layer, and it will increase its firing rate. So it's like an input of new information. And we have these inhibitory connections from deep layer interneurons to the superficial layer. And if we provide additional excitation to layer 5 due to these inhibitory connections, the gamma activity and also gamma band resonance will be suppressed in layer 2-3. And it will prevent input from affecting the circuit. Okay. And I used Dzykiewicz neurons. For excitatory neurons, I used models of pyramidal cells, regular spiking. For inhibitory neurons of layer 2-3, I used fast spiking cells, which are known to be involved in gamma band generation. And for deep layer inhibitory neurons, I used low threshold spiking cells, which are supposed to be involved in slow rhythmus generation. All of them are just parameterizations of the Dzykiewicz neuron. Okay, so the first step is to find the appropriate synaptic weights. So I delivered a single spike into each type of cells via AMP and GABA-A receptors, looked at postsynaptic potentials, and selected synaptic weights in such range that synaptic potential is about 1 millivolt, 1, maybe 2, like this. Okay, so of course we're able to model only a small network, which is embedded in a huge cortical network. So each neuron receives many external inputs. And when you have many inputs, each of them is weak, and all of them are asynchronous. This spike train, in fact, could be represented as white noise signal. I generated white noise signals, delivered them to AMPA and GABA A receptors. And to do this, I slightly modified this XPSIN module. I added a constant that is added to the conductance at each simulation time step. And then I load random noise into neuron vector, and I play this vector to this new variable. Okay, so the weights are taken from the previous step and are played with the firing rates of these inputs to achieve about 10 hertz firing rate and relatively high CV. Okay, so it is just a picture of how firing rate and CV depend on these two external firing rates, and this is the final choice. And here is an example of voltage trace. It's look nice. There are fluctuations, balanced excitation inhibition, and irregular spiking activity. Okay, and I did the same for fast spiking and low threshold spiking neurons. Okay, so now I started building a network. I took 200 pyramidal neurons, added recurrent connections with convergence of 50, and then to achieve the same firing rate as before, I decreased the external firing rate, which defines this white noise signal. And I did it analytically using this formula. And we can see that indeed in the connected and the unconnected versions, the firing rates are the same. Now I added fast spiking population. I added new excitatory connection, decreased the corresponding external firing rate, added inhibitory connections, decreased the corresponding external firing rates. So now we have these 4 expressions, which allow us to play with these 4 synaptic weights and automatically recalculate the external input, so we always have approximately the same firing rates. Yeah, this is the proof that the firing rates are unchanged. And here is the basic picture of the possible regimes of excitatory inhibitory network. And what we need is basically this one. So gamma rhythm, and to achieve it, the most important connections are between excitatory inhibitory and from inhibitory to excitatory. So this is the network with the original parameters, and after tuning these parameters, I obtain a peak in gamma band. Okay, and I did the same thing for layer 5, and here the peak is beta band. And now we have these 2 circuits. We have these peaks for them in beta and gamma, and now we connect them. Of course, decreasing the external weights. The peak for layer 2-3 is slightly shifted towards lower frequencies. And now the most important thing probably is we add an additional excitation to layer 5 with certain proportion to excitatory and inhibitory cells. And it destroys this gamma band peak in the spectrum. And instead of it, a beta band peak appears. And it has some functional consequences. Now, let us add gamma band input to layer 2-3 pyramidals. This input is supposed to be some new information coming to this circuit. And I deliver this during the second part of simulation. And you see that it indeed entrains the network. If I add this modulatory input to layer 5, we see that beta activity is increased. In fact, it is also transferred to the layer 2-3. The gamma entrainment in this case is not that stable. And if we look at the firing rate dynamics, these are firing rates average smoothed with sliding time window of 100 milliseconds. So we smooth out all these oscillations and only look at the average firing rates. And we see that in the baseline, this modulatory input doesn't affect very much. But when we deliver gamma band input to superficial layers, the presence of this modulatory input decreases response to the gamma input. And that is, in fact, what we wanted in the first place, because now we have a mechanism via which we can first increase beta power, and second, decrease sensitivity of the cortical column to gamma band input. And if we suppose that gamma band input is new information, decreasing response to this new information means that we promote the previous state, so cortical status quo. And here are the results, just a list of what I've shown to you already. And future, what should be done next is I want to find the parameters to stimulate the deep layer, not by tonic input, because in this case, this beta activity is in fact epiphenomenal. It is not directly involved in gamma suppression. But I want to stimulate this deep layer by beta modulated input. For example, we have such inputs from subcortical regions like subthalamic nucleus. Yes, so, and in this case, beta rhythm will have functional role. And of course, I want to make more realistic connectivity between layers, probably more realistic cells, which NetPyNE allows to do. So that's all basically. Thank you.
Thank you.
Great job.
Thanks.
Questions? And Afreen is going to be next. Yeah, I liked your parameter exploration. It was a nice combination of computational with analytical to a nice feedback and forth. I really enjoyed that. I don't know much about this topic, so I don't really have an intelligent question.
Nikita, you mentioned you were interested in looking at some of these oscillations in the detailed M1 model. These beta and gamma oscillations. Maybe you can say a bit about that. I thought it was like a very interesting idea.
I think that if you have a complicated, complex model of M1, it is important to really understand how these rhythms are generated, especially due to the fact that you did not optimize for those rhythms, that you get them like automatically for free. I think it is important. There is a, a paper on Potjans-Distman model about gamma rhythm, which tries to find these eigenmodes for gamma generation, because it is it turns out that it is not in a single layer, it is a multi-layered activity with certain frequency shifts and so on. And I think it could be interesting and important to do this with beta, to understand which circuit mechanisms are involved, which layers, which cells, and it is known that beta oscillations and other slow oscillations their mechanisms are more complicated than gamma generating mechanisms. And they involve specific cell types, like intrinsically bursting cells of deep layers, and also things like probably dendritic resonance. And there are two directions to take. So one direction is to try to disentangle these eigenmodes in the circuit network. For example, like take each cell, perturb each of its inputs with oscillation, with beta oscillations, for example, and then take the whole matrix of responses, and from this matrix we can try to calculate the response of the whole network and compare it with simulations. And another thing is to take each individual cell, deliver inputs to this cell, and play with different mechanisms to see whether we have beta resonance or whether we have lost beta resonance. So the basic idea is that, of course, it is impossible to resimulate the whole model each time. It is supercomputers, as far as I understand. But if we have a result of simulation, for example, spike trains, we can take them and we could deliver them to individual neurons, which could be simulated at a home computer. And basically do these two lines of research. One circuit and another is single cell with cellular mechanisms. So that's the idea. Okay.
Great, thank you a lot. I think that's really interesting.
Yeah.
We can talk more.
Okay, so I had Afarin on the schedule, but I don't actually see him in the room.
No, I think she's not going to present.
Oh, okay.